Hello, welcome to the proteomics course. In today's lecture, we will talk about applications of two-dimensional electrophoresis. In the last few lectures of this module, we discussed about two-dimensional electrophoresis technique. We discussed in much detail the workflow, how various steps are involved in performing two-dimensional electrophoresis and difference in gel electrophoresis. Now, today we will talk about how these techniques can be applied for understanding some of the biological questions. Now, this is time for us to assimilate all the knowledge which we have acquired during the last several lectures. For example, what are the things one need to take care of during protein extraction, then isoelectric focusing, second dimension protein separation, followed by gel analysis. So, all of this knowledge actually integrates when you want to apply these techniques in any biological applications. So, today I will just first give you an overview of 2DE technique, just to refresh you about various things which we discussed in this module. And then I will give you few example applications. We will take two case studies. Now, this two-dimensional electrophoresis technique is now widely applicable in almost all the areas of biological sciences. You name any application and you will see there are some published report on using two-dimensional electrophoresis or difference in gel electrophoresis to apply for such applications, whether it is looking at the human different diseases, looking at drug responses, or investigating about plant stresses, or any drug treatment in bacterial species or any other microbes. So, as I said, like you name any application and you will see that several published report where people have used these techniques and try to look for different type of proteome changes. Now, one can look at the differential protein expression, which is most commonly used if you want to compare a control with a treatment. You have certain level of exposure to a disease or a chemical or a drug, and you want to know what are all the proteins have been modulated due to that particular stimulus or treatment. Now, this is known as differential expression. Now, if you are interested to investigate all the proteins of a given sample, that will be considered in the global proteome profiling. So, many times if a proteome of a given species is not known, then proteomic techniques have been used for global proteome profiling, but most commonly they have been used for differential proteomics. So, today we will discuss two papers where authors have applied differential proteomic analysis by using two-dimensional electrophoresis. So, before I discuss the application, let us just refresh the whatever things we have discussed in the previous lectures on various gel based proteomic techniques. So, this slide reminds you that we have talked about different approaches used in the gel based proteomics, including SDSPH analysis, including two-dimensional electrophoresis, and various advanced level of protein electrophoresis, including difference in gel electrophoresis. Then data analysis, that becomes common for all of these gel based approaches, and that becomes actually very challenging aspect of it. So, performing a proteome experiment is not so tedious as compared to analyzing the large amount of data which is generated from these techniques, and most importantly, how to get some meaningful information from these experiments. Therefore, data analysis and statistical considerations are very important parameters for obtaining any meaningful information. So, first of all, you need to extract the protein from a given sample, and that actually becomes very determining factor for various applications, regardless of whatever questions you have in your mind, which you want to apply by using two-dimensional electrophoresis. First of all, you need to make a very good protein extract, and when I said very good extract, it means it should have minimal interference with any interfering components. Ideally, you want to separate as many proteins as possible. It means you want to get the very high coverage of whole proteome. So, considering the diversity of sample which one can apply on these techniques, it becomes very important that people first standardize very good protein extraction. Once good protein extract has been obtained and you have determined the quantity of the protein, then you can apply that on the first dimension, which is isoelectric focusing or IEF. So, in this you can separate the proteins based on their pi values as shown in the slide here. You can use different length of IPG strip or immobilized pH gradient strip and separate the proteins in the first dimension based on their isoelectric point. After first dimension separation, if you remember, then you need to do some treatment, which is equilibration. You want to prepare your strips for the second dimension separation. So, in equilibration, there were two components which were very essential, and if you recall, we talked about DTT and iodoacetramide. So, after doing the equilibration treatment, then you can separate the protein in the second dimension based on molecular weight of the proteins. So, now as you can see in the slide, you can separate the proteins in two dimension in two different properties based on their isoelectric point and molecular weight. This is a representative 2D gel image which shows that large number of protein spots can be separated on these gels, and immediately by looking at a given protein spot, one can obtain two information: what is their molecular weight and what is their isoelectric point. So, this information one can obtain immediately, and then after comparison of the different gels, one can know what are the proteins which are differentially expressed. Now, as I discussed in much detail, in fact, in almost a complete lecture earlier, that data analysis is a very crucial aspect of two-dimensional analysis experiment. You can run large number of gels, but when it comes to compare these gels, it takes a lot of time. You have to be very careful to obtain some statistically significant spots which could be meaningful for that biological application. So, having discussed the broad overview of the two-dimensional electrophoresis, now let us move on to the case study. In first study, we will talk about serum proteome analysis of vivax malaria and insight into disease pathogenesis and host response, a study by Ray et al. So, this study was performed by my group, and we have tried to investigate the host response against malarial pathogen vivax. So, if you look at this map of world population, so overall Asia and Africa are mostly affected due to malaria. Asia is suffering from challenges of drug resistant strains of malaria, and Africa is still the single largest cause of death, which is due to malaria. But not only in Asia and Africa, but malaria is an epidemic in 103 countries around the globe. And incidence of this disease causes more than 300 million per year and death almost between 1 to 2.7 million. So, this stats gives us an indication that how severe this problem is. And among various species of malaria, Plasmodium vivax and Plasmodium falciparum, these are the accountable for over 90% of malarial cases worldwide. So, the estimated annual number for the clinical cases of vivax malaria ranges from 70 to 390 million, and that is actually outside of the African contexts. Now, the P vivax accounts for more than 50% of all the malarial cases worldwide, but the morbidity associated with this infection and its spectrum of disease still remains neglected. Nearly 2.6 billion people around the globe are at the risk of Plasmodium vivax infection, even slightly greater than people who are at risk with the Plasmodium falciparum infection. Although the Plasmodium vivax infection is historically regarded as benign, but recently the severe and fatal incidence of vivax malaria has been reported from different regions of the world, which rendered this clinical paradigm quite deceptive. So, in the complex life cycle of malaria, now investigating how Plasmodium falciparum and Plasmodium vivax behave differently in human, and what are different type of proteins which are elicited due to the response against these pathogens. These are some of the very challenging questions in the malaria field. The slide gives you various species of malaria parasites which causes different type of malaria. Plasmodium vivax causes benign tertiary malaria, Plasmodium falciparum malignant tertiary malaria, Plasmodium ovale tertiary malaria, and Plasmodium malariae quarter malaria. In addition to these, Plasmodium knowleski can also cause acute and severe illness, but mortality rates are quite low for this species. So, there are different pathogens of malaria, but Plasmodium falciparum and vivax are the prime targets for investigation, because as we discussed earlier, more than 95% of malarial cases worldwide are due to these two species. So, this slide gives an overview of the workflow which we followed to investigate the host response against Plasmodium vivax in human serum. So, serum samples were obtained, proteins were extracted, and the protein samples were compared from the healthy and vivax patients by applying gel based proteomics, including 2-dimensional electrophoresis and difference in gel electrophoresis. We separated large number of proteins on the gels, compared the data, looked for those proteins which are significantly differentially expressed in vivax as compared to the healthy controls, and then those proteins were subjected to mass spectrometry analysis. Further, we validated few targets and analyzed the data for various type of pathways which could be involved in such disease. So, before we move to details of various experiments performed in this study, let me show you this animation to give you an overview of the workflow followed in this experiment. So, let me give you an overview of all the steps involved in performing such an experiment. So, first of all, carefully withdraw the intravenous blood into a vacuum inner tube and store the tube on ice to allow the blood to coagulate. Centrifuge the contents to separate coagulated blood cells and clotting factors from the serum, which forms a clear supernatant. After centrifugation is done, then collect the obtained serum in a fresh tube. sonicate the serum to break down any large protein complexes. Serum contains several proteins in wide range of concentrations, of which albumin and immunoglobulin G are the most abundant ones. The presence of such high abundance proteins can interfere with the experiment. Therefore, we need to remove these high abundant proteins. So, in this experiment we tried both sonication and depletion of the high abundant proteins. After sonication is done, then we can add the serum sample on the columns. Usually these are commercially available, which can deplete various high abundant proteins. Now, these depletion columns bind only the high abundance proteins onto their matrix through the affinity interactions. Once the serum has been processed using a depletion column, precipitate out the remaining proteins by using trichloroacetic acid and acetone. Once the protein extraction is done, then we need to add the rehydration buffer, which contains urea, thiourea, CHAPS, DTT and BPB. After overnight incubation, then we can centrifuge and collect the supernatant. Now, we will watch the quantification of the proteins. So, prepare the Bradford dye reagent and label the tubes suitably for the standard and test samples. Add the standard and sample solutions to their respective tubes. Now, add the Bradford color reagent to the tubes and mix them thoroughly. Once the color reaction has occurred, then perform the absorbance measurement at 595 nanometer. Once you have obtained good amount of proteins, which was determined by the protein quantification, now proceed for the rehydration of immobilized pH gradient strips and focusing of these rehydrated strips. So, remove the IPG strip from the cover, place it in the rehydration tray, add the reconstituted protein sample on the strip, pour mineral oil to prevent it from the drying, and then move forward for the isoelectric focusing. So, place the wicks at the either end of the IPG strip, followed by an electrode at each end. Fill all the adjacent wells with the mineral oil to ensure the uniform current flow. You can input the desired protocol on the instrument software along with the details of strip length, pH range, number of strips, etc., and start the focusing process. You can monitor the focusing progress on the software, and you need to ensure that focusing is proper. During the IF, proteins will migrate along the strip and come to rest at a point when their net charge becomes 0, which is known as their isoelectric point. After the IF is done, then we need to separate the proteins in the second dimension on SDS page. Remember, in between you have to do the equilibration strip as well. So, SDS page, which constitutes the second dimension of two-dimensional gel electrophoresis, involves assembly of the gel apparatus, gel casting, equilibration of IPG strip, followed by placement of the IPG strip on the gel and protein separation. So, prepare the gel casting solution consisting of acrylamide, bisacrylamide, tris chloride, SDS, APS and timid. Once the gel is polymerized, then you can add the IPG strip and now ensure that assembly is properly sealed and buffers are properly placed, so that proteins can be separated based on their molecular weight. Depending on the gel length, you can perform the second dimension separation for few hours. After second dimension separation is done, then we can perform the staining and de-staining. So, this process involves removal of the gel from the electrophoretic assembly, followed by treatment with a fixing solution, staining solution and finally, the de-staining solution. You need to ensure that proper shaking conditions are maintained, and you need to allow 10 to 14 hours, usually overnight step for the staining as well as de-staining steps. After de-staining is done, then you can see a protein spots on the gel with the very clear background. Okay, after watching this animation, now you must be clear of various steps which are required to perform this experiment. In clinical studies, first of all, obtaining the clinical samples and getting the right type of clinical sample, that is very challenging question. So, to obtain the samples, first of all, one need to do the ethics committee approval. Scientific committee has to look at the proposal and review and approve it, and then patients which fall into those categories which you want to investigate have to be enrolled after taking their consent, and ethically it should be approved. So, various type of information for such patient are tabulated, and then these patient sample can be compared for the proteomic analysis. So, in addition to the age, the gender, physiological status, whether patients have any alcoholic background, any previous history of diseases, or they have taken any treatment earlier. In addition to all this, various type of clinical tests which are performed, they are all tabulated, which eventually helps in analyzing the data when you want to compare the proteomic response for various patients. Selection of healthy controls become critical, because you want to compare healthy with the disease sample. Now, if you are healthy suffering from some disease, then that may give you some artifact information. So, that is why in the clinical studies you need large number of samples. Then there are issues whether somebody wants to analyze only individual sample of these clinical patients, or they want to pool it and then analyze. Consider you have 200 cases and 200 controls. Now, running individual gels for all of this number of patients and the healthy control can be very challenging, and then finally, running the gels in triplicate and analyzing the data, it becomes very tedious. So, there are discussions going on whether for such large number of clinical cohorts, whether group of patients and controls can be pooled and then analyzed. For example, rather than analyzing 200, if we make pool of 10 patients, then you have 20 samples. But that all depends on the investigators and the kind of experimental design which people have in their mind for performing such studies. Then sample collection process becomes very important. If you recall the previous lectures, how different type of storage conditions can also affect your proteome. So, all these parameters one need to take into the consideration. So, similarly, we also tabulated various information for different Vivax patients and healthy controls, this slide just gives you an overview that before even starting your proteome experiment, lot of thought has to go into your experiment, into your experimental design, and what type of samples one need to collect, and then you need to obtain various clinical information for those healthy controls and your patients. So, as you have seen in the animation, first of all you need to collect the blood sample, then serum has to separate it from that, and after serum is collected, then you need to store that in a small aliquots at -80 degrees. So, this slide gives you various steps involved in this process. So, ideally 5 ml of blood is sufficient for getting enough serum to perform such proteomic investigations. So, 5 ml blood if you collect and then keep it on ice until the serum isolation, allow it to clot for an hour and then centrifuge at 20 degrees for 10 minutes, followed by separate the supernatant, the serum layer immediately. Collected serum can be stored at -20 degrees or -80 degrees. -20 if you want to use that immediately for your further use, or if you want to store for long time, then you can store in -80. Now, serum poses challenge of abundant proteins. So, first of all we had to remove the abundant proteins from the serum, and there are standard columns available from various commercial manufacturing, from which one can remove selectively the very highly abundant proteins such as serum albumin, IgG and various other abundant proteins. In fact, there are columns which can deplete almost 14 abundant proteins present in the serum. Now, what should be ideal sample which one can use for such proteome experiment? So, first of all, to address this question, we tried separating the proteins from various type of samples. We took crude serum, because serum will have mostly the protein content. So, we thought, can we directly apply the serum on the IPG strip, immobilized pH gradient strip, separate the protein and see how many proteins we can separate on these 2D gels. But serum contains lot of salt. So, isoelectric focusing itself becomes challenging if you have crude serum. So, we also tried whether we remove the salt component of it, and after desalting if we do the IEF. Since serum contains highly abundant proteins and these commercial columns which can deplete these abundant proteins selectively, those are quite costly, and considering that large number of patient sample you have to process. So, we thought, can we disrupt these abundant proteins and remove these low abundant proteins from these abundant proteins by using sonication. So, effect of sonication as well as desalting was also investigated, and then we also used commercial columns to remove the abundant proteins followed by the desalting step. Now, sample was corrected and processed with all these 4 variables which I talked to you, and after that the standard procedure of 2-dimensional electrophoresis was followed. Now, when you are doing this serum protein separation, the IEF becomes very critical, because you have lot of salt component in it, and if your salt removal is not effective, your isoelectric focusing run may not be very smooth. So, from the softwares you can monitor how smoothly your isoelectric focusing is progressing. So, we tried various type of effects, crude serum alone or sonicated and desalted, desalted alone and depleted and desalted, and then try to compare the effect of these type of processing on overall proteome coverage. We also tried to see that how many protein we can separate on the small gels, and then as expected the small gel of 7 centimeter cannot provide us very large number of proteins, but even these gels can be used for standardization process, and as you can see, starting from the crude serum to the desalted and then finally sonicated and desalted gives us better coverage of the proteome. We tried the same with the even larger strip 11 centimeter, and it was again quite convincing. Then we moved on to the very large strips 24 centimeter for the clinical studies. So, in general we took 40 microliter of the serum, which was precipitated with the 4 volume of ice cold acetone containing 10% of TCA, incubated at -20 degrees, centrifuged, followed by added 1 ml of ice cold acetone to wash the precipitate. Then incubated it on the ice for 15 minutes, and again centrifugation was performed. Acetone containing supernatant was removed, and then pellet was dried in the lysis buffer. The recipe for lysis buffer we have talked earlier when we discussed about sample preparation, which is also mentioned in the slide here. You can use urea, chaps, IPG buffer, DTT and BPB. So, from the previous study we looked at replicate gels, and is 3 here, and we found that desalted, sonicated and depleted sample gave us largest number of spots on the gel as compared to crude alone or desalted alone or desalted and sonicated. So, these studies were performed on the small strips to standardize the procedure, but we are able to draw the conclusion based on the reproducible pattern obtained in these gels, and then we applied the desalted, sonicated and depleted these conditions for the processing all the samples. Now, after processing of these samples, now how to extract the proteins? So, protein extraction protocol was also optimized, and to optimize these protocols, first of all we looked at what are different methods available in the literature. So, people have applied direct crude serum, TCA acetone precipitation, sonicated and desalted serum, trizol extraction method, as well as abundant protein removal and TCA acetone precipitation. So, we used this modified procedure of TCA acetone precipitation, where we used depleted and desalted and sonicated serum. We also compared the effect of acetone alone or TCA acetone precipitation. As you can see in this slide, the left one is showing the overall protein spot present in the acetone alone treatment, and right side slide showing the TCA acetone precipitation. So, overall more number of spots were resolved on the TCA acetone precipitated gel. So, since we standardized our condition on the small gels earlier, we also try to apply that on the large gel of 24 centimeters, because that is where we have to perform all our analysis. So, we again compared crude versus depleted protein response of these serum samples on the large gel, and as expected the depleted serum sample showed more number of spots as compared to the crude serum samples. We also try to look at effects of various type of staining, whether it is Kumasi blue staining or silver staining. We definitely identified more number of spots from the silver staining, but just because of we want to compare a large number of clinical samples with the different healthy controls, it was very difficult to standardize the conditions to keep it uniform staining for the silver. So, to overcome this limitation, we use the colloidal Kumasi and biosafe Kumasi staining, and we standardize the same condition for all the gels. So, this slide gives you an overview of various type of parameters which we investigated from different type of treatments: crude serum, sonicated and desalted, TCA acetone precipitated and trizol extraction methods. So, we looked at effect of sonication, desalting, rehydration, amount of protein loading, type of strip, staining solution, how many number of spots we can resolve on these gels, and then since software analysis automated gives lot of artifacts, how many real spots we can obtain after the manual refinement. So, I am trying to give you all the details what one need to actually perform before reaching to that stage where you can compare the healthy control with the clinical samples. And this lecture is actually trying to cover various type of concepts which we have talked in the sample processing and in different lectures of two-dimensional electrophoresis. So, now after doing lot of comparative analysis and standardizing the protocols for sample preparation and protein extraction, now we are ready to perform the comparative study on serum samples of Vivax patients with the healthy controls. This is one of the gel image showing that is what is expected on 24 centimeter large 2D gel, and then a representative gel showing the healthy control and the Plasmodium vivax treated samples. So, these samples as you can see were showing large number of protein spots resolved on these gels, and then after separating these protein samples on these 2D gels from various patients and controls, then most challenging task was the data analysis. So, automated matching as well as manual refinements are performed to obtain the how many spots are significantly modulated due to Vivax infection. As you can see in this slide, we had looked at various controls and various Vivax patients. These are only few patient samples here, but we analyzed a large number of samples, but those spots which were reproducible in all the patients. For example, one you can see in the top panel, 2 spots are showing significant alteration in the Vivax as compared to the healthy controls. So, these type of spots were considered further for the mass spectrometry analysis. Now, those spots which we found were showing good differential expression, we looked at the 3-dimensional views of those spots to ensure that those are not artifacts and those are the real spots showing up or down regulation of these proteins in human serum. Now, after completing the analysis, we were able to obtain large number of proteins which were differentially expressed, and as you can see in this graph, there are proteins which vary in different range of the fold change. Many proteins show very less fold change between 1 to 1.5 fold, and there are few proteins which also show more than 10 fold up or down regulation. So, these protein spots were considered interesting because those were statistically significant, and then these spots were further analyzed using mass spectrometry techniques. So, this slide gives you an overview of tandem mass spectrometry technique and followed by how LC-MS/MS data can be analyzed by using mascot search engine. So, different type of mass spectrometry techniques are available, including multi-TOF and LC-MS/MS based approaches, and we will talk about these techniques in much more detail in the next module when we talk about mass spectrometry. But just to give you an overview here, that one need to analyze the spectra and also one need to have some standard proteins to ensure that correct spectra is obtained. So, you have to do a lot of quality control checks in mass spectrometry to ensure that your data is of good quality. I will cover the details of mass spectrometry experiment when we talk about mass spectrometry in more detail in the next module, and again maybe I will take this study at that time, that what are the different detailed procedure required to perform such analysis. But at the end, from these experiment of mass spectrometry, one can obtain the identity of these proteins, and as you can see in the slide, there are several proteins in the host human where differentially expressed due to the Plasmodium vivax infection. And these are some of the proteins listed here, such as haptoglobin, apolipoprotein A1, serum albumin precursor and clusterin. These were down regulated, and serum amyloid A, celloplasmin precursor, leucine rich alpha 2 glycoprotein precursor and alpha antitrypsin precursor. These proteins were upregulated among many other proteins. So, conclusions from this case study, that few differentially regulated serum proteins were identified in this study, including celloplasmin, hemopexin, alpha anti-chymotrypsin, alpha 1 antitrypsin, which have not been previously reported in the vivax malaria pathogenesis. A very important role of few proteins such as serum amyloid A and haptoglobin, apolipoprotein A1 and E was established in the vivax malaria, and this study finally enhanced our understanding for the basis of disease pathogenesis and provided few potential targets which could be used for further investigation. Today we talked about how two-dimensional electrophoresis technique can be applied for various biological applications. Although I thought I will be able to cover two case studies on malaria, one on the host side, like how host response can be monitored due to the infection of Plasmodium vivax, and then second study which I was going to take was based on the effect of a drug on another Plasmodium species, Plasmodium falciparum. So, we could not discuss the other study on the drug treatment of malarial parasite, which we will discuss in the next class, but by giving you a case study and details of various type of experiments one need to perform, probably you got an idea that before performing that final experiment on your case and the controls, a lot of optimization has to be performed, and starting from your sample processing and how you can expand the coverage of the proteome. These type of quality control checks are very essential. If you can increase the overall proteome coverage, you can separate over 1500 to 2000 proteins on the gel reproducibly, then you have a good chance of identifying various potential targets, which could be novel drug target or potential biomarkers. But if you have not done the proper quality control experiment, you have not optimized the protocols properly and your proteome coverage is very poor on the gel, then you are comparing only partial proteome, and there is a good likelihood that you will miss out many important changes. So, not only today we discussed about one application, how host serum protein changes due to one of the Plasmodium parasite, but also we discussed the various nitty-gritty experiments one need to perform to achieve such type of comparison. We will continue our discussion on some more applications of two-dimensional electrophoresis and also two-dimensional difference in electrophoresis, because due to the variations in the 2DE, people also try two-dimensional Dij experiments, and also that is very sensitive. So, that also expands the overall proteome coverage. So, we will continue our discussion in the next class. Thank you.